Alright, and welcome. So we're going to talk about the history of fish, which is really the evolutionary story of fish. And the evolution story of fish is, I think, a very interesting story, partially, again, because we are a derived group of fishes, but also partially because many of the themes that we deal with in evolution are writ large in vertebrate history. And for vertebrates, actually, we have a relatively good fossil record, so we can actually really get a chance to look at how evolution proceeded with these groups. It doesn't mean that we have fossils for every point that we would like to, but we have so many fossils now that we have very, very good understanding of how evolution actually proceeded in a lot of these different groups. So what we're going to do in this lecture is we're going to talk about how fish evolved, and traditionally this is saved until we go through some of the physiology of fishes, but I actually think it's important to get an understanding of how evolution proceeded, and then we'll go back and talk about the physiological mechanisms and how those were actually picked up along the way. Remember, what you see today in the form of something like the fish on the left here that are pictured is a very, very derived version of what first evolved within the vertebrate group. And I want to stress again that this is usually considered middle of the semester sort of material, but I really don't think it makes any sense to talk about biology without starting from the concept of evolution. Evolution is the foundation upon which all other biological sciences are built, and so it's really important that we understand how fish evolved, and then we'll talk about why they might have evolved in that way. So again, when I use the word fish right now, what I'm really referring to is all vertebrates. Remember that the distinction between what we consider fish and sort of an aquatic animal and terrestrial animals is not a real distinction. They're one and the same. Terrestrial animals belong to the group of lobe-finned fish. So they're actually all fishes. What we're going to do in this lecture is we're going to sort of travel back in time. We're going to need to go back to the initial phases when vertebrates were first evolving, and then we're going to sort of step through time until we come to modern groups, which have and continue to evolve as all groups that are in existence do. And those groups that you see will look very familiar to you, but some of the groups that you see initially, which will be what we call the primitive groups, will look very, very unfamiliar to you. They won't look like much that you recognize. But I want you to understand that when we look at those groups, the things that make fish fish are present in those groups, but not in the same ways that they will be present in future groups. And I don't want you to get confused or bogged down on what that actually means right now. Once you start to see how the fish change through time, it'll make more sense. So what we're going to have to do is really travel through time, and we're unfortunately not going to be able to get into a time machine to do this, but we do have the ability to imagine what happened in the past, and we also have, again, a fossil record to guide us. So using both our imagination and what we know, we're going to go back in time and really think about what that actually means. The one thing I will mention for people that have older textbooks is that the history of fishes is constantly being updated, and I would say that it changes almost weekly. That doesn't mean that the whole picture is thrown out every week, but it does mean that on the story of the evolution of fishes, many details are changing all the time. We know the large picture, and we've known that for a fairly long time, maybe on the order of hundreds of years, but the individual picture of how individual groups are related to one another, that's something that's constantly in a state of flux. So with some groups, we're very certain where they belong. With many other groups, we're not quite certain where they belong. So if you have an older version of a textbook and you save money by buying that, that section won't be uninformative, but it won't be nearly as informative as the most recent version. Keep in mind, though, that these are all textbooks, and the moment that they're printed, they're already out of date. And that's certainly true within any rapidly moving field. If you buy, let's say, an immunology textbook, usually before you've even cracked the book open, the book is extremely out of date in various portions of the textbook. And you could actually highlight almost large sections of chapters and say these probably need to be rewritten at this point. But for the history of fishes, we would still have to go back and change a lot of stuff, but there is still good information in older versions, but the relatedness of groups to one another was not well understood, and so you'll see that if you go back and look at older versions of the textbook and compare it to something like this lecture. So where are we actually going to go here? Remember back in the first lecture I showed you this, what would be called a cladogram. It's a tree, a branching tree of life and the relatedness of fish to one another. And also here, if you look down at the lower left-hand corner, non-fish, right? Sea stars and acorn worms are really not fish in any sense of the word. So those are related to us in some way, but they are not fish. What we really want to do is start to understand what these groups look like, what these branch points actually look like, and as well as why these branch points exist. And the why we're not really going to get to in the history of fish, because that's really going to talk about how physiological mechanisms evolve, why physiological mechanisms evolve, how ecologies evolve, why they evolve, and that's a topic that we need to delve into in a separate lecture. But I didn't want you to be shown a bunch of fish and just say, here's a bunch of fish, you may know nothing about them. We're going to just talk about them, about their physiology, as if you would have some inherent understanding. I want you to at least have an image in your mind when we talk about these animals of what these animals look like and how they might have behaved, so that you can start to understand why they might have the ecology that they do. So we're going to go through these fish. Today we're actually just going to deal with the jawless fish, which leads us up to, where are we at? Up to about here. We're going to go through these animals down here. Jawless fish, relatively speaking, are mostly extinct. So most of the animals that we'll talk about today are no longer present in the... Before we get into the world of fish, we're going to have to step back to the world of geology and the world of ancient time, and what we call deep time in many cases. This is the history of the Earth, broken up into different parts. The part that we're actually in right now is the Cenozoic. The Cenozoic is the all of the time since the last major extinction event, which was the KT boundary, which is where most dinosaurs go extinct. You can see that down here at the bottom. It starts at 66. Dinosaurs go extinct largely at 65.5 million years ago. So the Cenozoic is the start of that age. We're all the way at the top, right? So we're all the way at the very top in the Quaternary. Actually, the Holocene, which was the epoch that we were just in, has been relabeled now. I think that we have a new epoch right on top of it, which is the Anthropocene. But I can't remember if that's been widely accepted yet or not. But in any case, the Cenozoic will have mostly modern fish. You will be very comfortable even at the very early portions of the Cenozoic down here at the very, very dawn of the Cenozoic. You'd be very comfortable with the animals you'd see. They'd look a little old to you, a little unusual, but you would be able to identify them, especially after you go through this class. If you go back further in time, you'll see that that won't necessarily be the case. If we step back again, this will take us from that 65.5 down to about 250 million years, which is nice. This is a Mesozoic, and the Mesozoic is frequently called the age of dinosaurs. That's probably true. It can also be called the age of reptiles, which is probably a fairer distinction if you're dealing with terrestrial animals. The Triassic, the Jurassic, and the Cretaceous. And of course, people know the name Jurassic from Jurassic Park, but these three periods make up the Mesozoic era. And the fish that you would see here are a little more unusual. You probably won't recognize a lot of these groups, and they will be, there's actually a larger diversity in some ways within the Mesozoic. If we travel back even further, so from about 250 million to 540 million, which is quite a long time ago, we end up in what's called the Paleozoic. And the Paleozoic really starts down here at the bottom with the Cambrian, when the first animals really start to appear onto the scene. Prior to that, almost everything in the world is probably single cellular, or at most colonial, meaning that there's maybe hundreds to maybe thousands of cells. During the Cambrian, what happens is we have the Cambrian explosion, and what we get is really complex organisms, including our own group, the vertebrates, will appear within the Cambrian. And then if we were to travel back even further, there's no fish in the Precambrian. The Precambrian, again, it's either all bacteria and or it's nothing. When you get really far back into the Precambrian, what you end up with is an Earth that's so hot that there's nothing living on it. It's just a molten surface, right? As you move into younger periods of time, and younger periods of time here are still pretty old because we're still dealing with billions of years, right? This scale is from 540 million to 4.6 billion years ago. So this is a very large period of time. Again, most of the stuff here is probably a surface of the Earth where it's so hot that nothing can grow on it. That you can see things like the Hadean period, right, which refers to, using the Greek here, hell, referring to the very hot surface. But the Proterozoic is the period at which we really start to see prokaryotes, bacteria, and that kind of stuff growing. And this is a period where life is really thriving, but it's life of a single cellular state, not life as we would think about it as a complex multicellular animal. So the foundations for fishes will appear in the Precambrian, but fish themselves will not really appear until just prior to the Cambrian, at about 540 million years ago. So this is a point at which we think somewhere within this realm, this star covers a lot of time period, right? The star right now is covering more time than humans have existed on the Earth, far and away. In fact, this star covers more time probably than the great apes. That would be the entire ape lineage has been on the surface of the Earth. So it's a broad area where we're not quite sure where that happens. But the issue, of course, is the further back in time you go, the harder it is to find fossils, and also the softer and smaller the animals are, and the harder it is to actually track them through time. But the genetics are telling us, yes, they evolved very, very early, but they probably really didn't look like much until much, much later. So how do we actually get to fish? Well, it turns out that animals, again, evolved sometime in the Precambrian. Everything would have looked something like a worm in a sense. It would have been a kind of a long, skinny animal with some sort of differentiation at one end, with something like cells that look something like a brain, that there's a gut that passes through it, that there appear to have been eyes in the very earliest animals as well, or at least photo-detecting spots in those animals. And that seems to be centralized around some area. And then, of course, there's another end, which is the tail and the anus, and that seems to have been set in animals relatively early. Most animals belong to this group on the left here, the protostomes, which is a group of animals you can see here that go through a very specific type of embryology, and it really matters here when the coelom is forming. The 8-cell stage of the protostome is a little bit different from the deuterosome, which is what our group belongs to. But really what's important is the coelom formation, where the mesoderm, which is one type of cell or one type of material that will make up different organs in the body, the endoderm and the ectoderm are actually arranged slightly differently than in the deuterostome. The interesting thing, of course, is that as you proceed through to what here is called the fate of the blastopore, which is just a slightly more advanced step in the development of that embryo, is that they look identical, right? After some very early initial steps where they look a little bit different, not much, but a little bit different, they end up looking identical to one another. And these will develop into the various animals that their genetics code for. But there was a divide at some point where a group of protostomes started to do things a little more like a deuterosome, and then eventually this early development for deuterosomes evolved, and that is the group that led to what we think of today as fish. And this group actually is a broader group, and this is how we know that we're related to other things. Deuterosomes are not that large a group, and it's a very specific way in which development proceeds, and those groups that proceed on that pathway are all related because they must have had a common ancestor at some point that proceeded along this pathway. One of the questions, of course, that humans would be very interested in answering is, well, why should there be a difference? Why would it matter if you end up looking identical? What's the advantage of forming the mesoderm slightly differently in one group than another? And the answer to that is that I don't know, so you can't ask me that. But it would be interesting to know. I don't know that there's actually a clear answer yet why this is the case. It could be something as simple as random chance that caused mesoderm not to fully separate from endoderm, and then it ends up actually being hard-coded into the development pathway to prevent large issues with development of organs further on. But it could also be that there's an advantage or a disadvantage to doing things the way that deuterosomes do or that protozoans do. Usually there's trade-offs when we see changes in development. In any case, these lineages probably divided broadly around 600 million years ago, so before the Cambrian. This is, again, relatively early before the Cambrian, but the world at that point would have been a very different world. The world would have been a very murky ocean that would have been filled with bacteria primarily, and there would have been relatively little oxygen in the ocean itself except in the top layers. And the surface of the Earth would have been, again, devoid of plants and animals, and certainly, and that would be very unusual to you, right? So if you walked around on the surface of the Earth, you would see bacterial films on that surface, right? You would see these sort of thick, gooey mats around wet areas. If you walked elsewhere, it would look like the worst kind of erosion you had ever seen in your entire life. There would be giant rocks exposed. There really wouldn't be soil. And whenever it rained, there would be sort of flash floods, and they could be potentially very catastrophic if you're living there. So the surface of the Earth would have been a very weird place to have been. You would have been able to breathe, actually. There would have been enough oxygen for you at that point, but you would not have recognized it as the Earth that you know today. In any case, so that happened maybe, let's say, 600 million years ago or somewhere around there. Really don't know. Again, finding in the fossil record an embryo of a deuterosome would be very difficult. You would be looking for something that is 8 cells large or maybe slightly larger than that, and you would be looking for something that's not really going to preserve very well. So unless you have very, very, very good preservation and those rocks have managed to survive and they've managed to weather to the surface, and someone who is lucky enough to just pick one up and say, you know what, I'd like to look at this under a scope, you're not going to have any fossils of those animals. So those kind of fossil records are very hard to find, unsurprisingly, and as a result, our actual timing of that event is hard to really nail down. And we may not really know that. I won't say ever, because every time someone says ever, it seems like somebody figures out a way to look a little bit further backwards. But it will be very difficult. It will be one thing that will be very, very difficult for biologists to do. But let's move on. So deuterosomes are the lineage that will give rise to the fishes and more importantly to things that we're concerned about like us. But let's start at the bottom of the deuterosome. So this is a branching tree. This is a very simple cladogram. But all of the animals that you see right now are deuterosomes. Echinodermata, which are the starfish. Hemichordata, which are the acorn worms. Acorn worms are not particularly attractive animals, but you would be surprised by how much they actually, at a biological way, that they actually look like us. They're really set up to look like other deuterosomes, and they start to bring to the table things that we think of within other lineages. For instance, Hemichordata really have what look like a primitive kidney. It's a very primitive kidney. It doesn't really look like what kidney you'd think about if you got a kidney out of an animal that you're going to eat, but it does some of the same things, and it's set up in the same way. And that, again, really speaks to the close relatedness of this group to us. It's not that Hemichordata doesn't have a good kidney, it's that it has a function, a group of cells that function like a kidney well enough for it that it doesn't need a better kidney. And then, of course, if we were to fall off this tree right down here at the bottom, we'd end up back at the protosome. So here are the deuterosomes. At least the starfish should be relatively familiar to people. You see here eventually develop into the chordates. And the chordates are a very, very, very advanced form of that larvae. What's actually happened now with the chordates is that the body of the larvae has been drawn way, way out, and in so doing, that's required that the body use muscles to actually move the body. Because the body is so much larger, the little tiny cilia will no longer propel it through the water column. Muscles are certainly present in things like Hemichordata and Echinodermata, but the ability to move the whole larval body via muscles is something that chordates will pick up. And chordates are really going to start to look like our primitive fish. And this right here is what we think of when we think of a very primitive classic chordate. Again, we have fossils that look similar to this. This is a way to to give you all of the characteristics that make a chordate without making it too complex. But these fossils are very rare, right? You're dealing with animals that are going to be very, very small. They're only going to be slightly larger than a very minuscule planktonic larva, and they're only going to be soft-bodied organisms. You don't have any of the hard parts yet. Chordates does not mean bones. Chordates really refers to these 5 characteristics. There are muscle segments, which are called myomeres, and they're shaped into these chevrons, these V-shaped things, and you'll see that in fish, right? If you dissect a trout or something, if you were to fillet off a fish body, look at the muscles. They are arranged in these chevron patterns. They are actually chevrons, they actually look like W's, but that's because the chevron has folded yet again. If you look at very small larval fish, what you'll see is they look a lot like V's, and that's because we're chordates, right? We bring all these characteristics with us. We don't just get to dispose of them at will. We have to bring all the steps that that we went through with us in some ways. We have a notochord, which is a cartilaginous tube that runs immediately underneath, or what we say ventral, to the dorsal nerve, hollow nerve cord, which is also a characteristic. Your spine is composed of bone, right? But in between the actual spinal discs, right, those things that support and protect your dorsal hollow nerve cord, which you do in fact have, are these very soft, spongy things that are called discs, or that are called discs, and that can be popped, and that can also be damaged. And that's actually your remnant notochord. What actually happens is the notochord is cartilaginous, and as your bones develop, most of the notochord degenerates. But to prevent the bones from sitting directly on top of each other, a little bit of the notochord remains, and that allows flexibility in your spine. And that's true of fish as well. They also keep, usually, they keep a relatively small amount of notochord. In primitive groups, the notochord is far more than sufficient to support the body and the muscle activity. But as we get into more and more muscles and larger muscles, you start to need more and more support elements to allow the muscles to pull, because small notochords will literally tear apart if muscles get too large. But if you're if you're 1 millimeter long, you can get away with a notochord very easily. There's well more than enough strength in a notochord to support you in that way. If you go down into primitive groups like lampreys, what you'll actually find is that they don't have those bones, right, the vertebral column, and they do rely entirely upon a notochord, right, to support their movement within the water column. The other thing that all chordates have is these pharyngeal slits. Pharyngeal slits are just slits on the side of the esophagus that allow water to be expelled, but that usually have some sort of filtering mechanism that allow particulate to be captured. So what we're going to see in primitive fishes is they're not going to be animals with jaws that go out and actively hunt things. They're going to be small, they're going to be relatively pathetic looking animals, and they're going to spend most of their time swimming around in the water column, filter feeding on plankton, which is probably going to be things like bacteria and algae, right? Those are the things they're going to be doing. And then the other really important thing for our group to note is that our group is one of the only groups that has this. And in fact, this in conjunction with the others makes this group what it is. We have a postanal tail, which means the tail extends beyond the anus. In most groups, the anus ends at the very, very apex of the body, right down here at the bottom. It'll extend all the way down to this point in the body, and it will terminate there at the end of the tail. Vertebrates, on the other hand, have pushed the anus further back into the body, and they allow the tail to extend further, and that allows you to fill the tail up with muscles, right, and make a very powerful tail while condensing the visceral organs into one protected area. Okay, so this is the chordate body plan. We're going to enter into the chordates now. We've moved out of just deuterosomes, and we're going to see the chordates. And the classic chordate would be the CEPHALOCHORDATA, which are sometimes called lanceolets. And lanceolets are more derived, again, than the echinodermata and the hemichordata. We're going to look at a close-up picture so we can see those characteristics very closely. And these animals look a lot like primitive fish. In fact, they look so much like primitive fish that historically people have placed them very, very, very closely to the branch between chordates, all other chordates, and fish. And that is it turns out not actually true. It's convergent evolution that they look so much like fish. but they do look a lot like fish. It turns out our closest living ancestors, for a variety of reasons, belong to another group we're going to see in just a bit. But let's first look at the Cephalochordata. Again, these are called lanceolate sometimes, or Amphioxus after the genus name of one of the groups. Pretty rare animals, not particularly common, always found in marine environments. And they spend most of their time like this. They are burrowed, they have their head poking out, and they have some what are called tentacles, which filter the larger particles of food away from, or keep larger particles of food away from the inside of the mouth. Water flows into the mouth. Most of the water passes out through the pharyngeal slits. A little tiny bit of water and the food gets sent down into the intestine, and that gets processed and then spit out at the anus. Note here we have the postanal tail. We've got a notochord, and we have a dorsal, you can't see it in this cross section, but it's hollow. This is a dorsal hollow nerve cord right here. Interesting thing, why are these things, how can this be a fish? Why would this even remotely look like something like a fish? Well, keep in mind, there are a lot of things that look like fish here, and there's a reading associated with this that will, I think, really convince you that the characteristics that are within the Cephalochordata really make it look like this slow sequential addition of different characteristics that make vertebrates in the future. One of the things I'd just like to point out is, look at this bulge right here, right, in the intestine. This bulge is very weird. It's a blind pocket, so you're not really going to send food into there because then it would have to just come back out. But what does this actually do? Well, this secretes enzymes into the intestine, right? It is able to spit out enzymes in the intestine. Well, where do we even have something like that in vertebrates? Well, that's what the liver does. And the liver is derived from the same tissue that makes the intestine, right? So how does the liver form? It forms from an outpocketing of the intestinal tract that seals off and then derives into liver. So this is really starting to look like a very primitive liver, right? It's starting to do things that we think of in other groups now, and those characteristics are being picked up very slowly through time. And there's, again, lots of other characteristics that make Cephalochordata look a lot like a very primitive fish, and that's really exciting. That means that our intuition that this animal is closely related at some level to us is true, and also that the steps that lead to fish that we would have to guess at, we can look at this animal and say, well, if it's present in this animal, it may very well have been present in primitive fish, and we know at least what to look for. Again, things like this are still clearly, if you picked one up, you would call this a, quote, invertebrate. Invertebrate is not a real group of animals because it encompasses so many different branches of the tree of life, but it is not a vertebrate in the sense it certainly does not have vertebrae. So let's talk now about what we think is, in fact, our closest living relative amongst the non-vertebrates, or the non-fish in this case, and that would be the tunicata. Tunicata are sea squirts. They're actually named after tunicates because they look a little bit, are tourniquets. They look a little bit like the things that you would wrap around an arm or a leg to prevent bleeding, and they're a little bit weird in appearance as adult. But again, if we look at the larvae, we see something very different. This starts to look a lot like a fish. We have a long postanal tail. There is a notochord that runs back here. There is a dorsal hollow nerve that runs back here. And the animal swims actively in the water column. Interestingly as well, the Cephalochordata spend most of their time burrowed in substrates, and they rely mostly for feeding on cilia. Right? They don't really swim around looking for food. They bring water in very slowly with cilia that beat water in. They're relatively small. They can get away with that. As you get into larger and larger animals, you can't get away with cilia. They're too small. They can't move enough water. Well, tunicates also rely actually on muscles to pull water in. And so in this way, they're very, very different than Cephalochordata. And this is how fish do it, right? Fish use muscles, a very muscular pump, to pull water in across pharyngeal slits where they filter out food or water and expel the water and then collect the food. And that is common to all fish in aquatic environments. In terrestrial environments, what's actually happened is that the lobe-fin fish, and that would be our lineage and all other terrestrial groups, have sealed off the pharyngeal slits. And usually they have a way of expelling water if they want to, but they tend to take food in that doesn't have nearly as much water associated with it, so they're not terribly concerned with the pharyngeal slits basket being available, which in this group is present right here, right? It pumps in water, expels the water, and then collects the food in that before sending it on to the intestine. The other thing that's interesting to note about this animal is it's really starting to condense the visceral organs into one place. It's able to say, look, it's able to move all those visceral organs into one place, and as a result, it can focus areas of the body into really active muscular sections without worrying about bringing the problems of having active muscles around organs that you may not want deformed, right? It's not really that great if every time you move, you change the size of your intestine, because that means that if you eat a large meal, you can't move. That's pretty risky if you're trying to get away from something. They are relatively small. They're not huge yet, but they use muscles, and they are filter feeders. And these are relatively active midwater animals. These are animals that swim around in the water column. So even though these guys may not look immediately like our most common ancestors, they act a lot like them. And the genetics say, yes, very closely, these are related to you. And that seems to make sense when we look at them at a very close level. So now we've crossed from what are not fish into the true fish. And the true fish here I've called CYCLOSTOMATA, which are the group that includes modern-day lampreys and modern-day hagfish. And these are relatively interesting groups because of the fact that they're so unusual from a lot of our other fish, but they appear to be relatively good examples of primitive fish. Now keep in mind, what did not happen in evolutionary time is there was not some actual line that divided the fish from the non-fish. It's a gradient, right? Just as you did not reach some age and immediately become an adult, it was a gradient from a child to an adult, and some people arguably remain children their whole lives. But the point being that you have a gradient of growth. And there's not some magical birthday where you become an adult. For purposes of taxes, there is a magical birthday where you become an adult. But for the purposes of actual development, there's not some magical moment in time where you've developed into an adult and you take on all the responsibilities of an adult, and there are horns playing in the background and the light shines and you say, I'm an adult now. And that's true of fish as well. It's not that there was some magical day where there were horns playing and there was someone on a piano in the background saying, and now we have fish, and a fish appears on the surface of the earth. That's not true. What happened is all the characteristics that we think of in a fish slowly appeared in groups, and at some point there were a lot of groups positioned very close to this fish break-off point, and one of those groups developed into what we consider as the modern group of fish, and the other groups went extinct. Right? We have one group that gave rise to all other what we think of as fish, but the odds of you finding that group in the fossil record are basically zilch. There were literally many thousands, tens of thousands of species. In the history of the Earth, we think that in the current Earth, we think that there are about 1% of all the species that have ever existed on the surface of the Earth right at this moment. So finding the species that would have given rise to all other groups of fish is hard to imagine. Besides the fact that the vast majority of species never get fossilized. So what we do is we sort of look at these branch points and we say, well, if the characteristics in these branch points are relatively similar to one another, we would expect those to be similar at all of the branch points, right? If the characteristics in Cyclostomata are similar to what we see in other groups, we would think that the last time those guys shared an ancestor, both those groups had that characteristic. But anyway, the Cyclostomata are the modern fish, and this is really exciting because we're into the real fish now. But these are jawless animals. They won't have paired fins, but they will rely on muscles, and they're going to get much, much bigger than the small animals that we've seen before, but not initially. They'll be relatively small. So again, let's go down to that branch point. Let's say we don't quite know if this is a fish or not. This is PIKAIA. PIKAIA appears in the Cambrian, so about 540 million years ago. It's swimming around. It's definitely related to us in some way. You can see that it has this very muscular body, but it basically looks like a tunicate, a flattened out tunicate with a more clearly defined head. This is a relatively unusual animal. But it's not clear exactly where it falls. It may be a fish. If it has vertebrae, it really becomes a fish, or it becomes a chordate, but it may just be but it may not. It's very hard to say. Fossil records of these soft-bodied animals are important, and you can see a fossil up here in the right-hand corner, but they don't let us know as much as if we had the physical animal in hand and we could dissect it. Again, this animal is relatively small. 5 centimeters long is a very small animal. That shouldn't surprise you. You know the ancestors of fish are probably basically small larvae that spend more and more time in the water column and less time on the bottom sediments, and as a result, they're just big larvae in this. Another fish from very close to that. So the last one was found at 530 million years ago. This one is at about 525, so it's slightly younger. It's about 5 million years ago. Or 5 million years closer to our time, which doesn't seem like a lot, but actually in the scope of evolution, it appears like a lot of stuff is going on. What you could see is it's something like a fish, right? We're not really sure. It might be a fish, but we need more specimens, and we need to actually figure that out. You can see a postanal tail in this fossil. I like this drawing a lot. You can see a postanal tail. The anus is going to come out further down. But you can see this tail over here extending out of the body. You can see those chevron V's. You can see a notochord actually in this fossil, and that would mean that there's a dorsal hollow nerve cord above it. And you can see the mouth. You can see this head develop. You can see lots of gill pouches, which means that there's a pharyngeal slit. And you can see that this animal looks like something that would be relatively active. This is probably, again, another very small animal, 2.5 centimeters. That's 1 inch for people that are using the English system. But it's swimming around in the water column and actively filter feeding, right? And that's something very different from what we've seen before. This appears to be a true craniate in that it is, in fact, probably a real fish. It has real developed skulls. But we're not really sure. It's hard to say. Unfortunately, the fossils often have damage to the head region, and the head is what we really need to see to know if it's a craniate or not. But for the moment, let's say it's probably something like a fish, and it certainly looks a lot like what a primitive fish would have looked like. The colors here, we don't know if the colors look like that, but this is a pretty good guess, actually, based on what we know about the colors of marine animals. We would expect to see colors like this in this animal, and certainly colors have been present in animal lineages for a long, long time. Relatively speaking, though, this animal's pretty far down on the food chain. At 2.5 centimeters is a filter-feeding active worm in the water column. Probably everything that can is eating it. So that's just too bad if you belong to a primitive fish. If we go into what we know are what I would say, yeah, so most people would identify these as fish-like, and these certainly, at least by our definition, fall within fish. We end up with the 2 modern groups. Again, these are all jawless. You can see down here that there's no jaw. It's just a disc. It can have teeth in it, although these teeth are not the same as your teeth. And these groups, there are two large groups within here. The hagfish, which is this one on the top, this pink worm thing, and the lampreys. It turns out, actually, there was a lot of debate whether this is one group or whether it was multiple groups. And hagfish seem to have a lot of characteristics that make them look very primitive, not least of which is that they do not regulate the ions in their body. So they have the same salinity as seawater. And that's pretty unusual with invertebrates. Very, very common in invertebrates. In fact, the most common strategy in invertebrates is to be the same concentration as seawater. But for vertebrates, very unusual. Not something you see. And so there's a lot of debate whether these 2 groups were related, and that seems to be settled now. These are in fact very closely related. Hagfish are deep water animals. They seem to have been under very stable forms of selective pressures for so long that they have radically changed the vertebrate body plan. In fact, and it looks more primitive than it actually is. It's actually derived condition that you see in hagfish. Lampreys are far more like the fish that you think of. They regulate their body. They have these very clearly defined eyes. They have pigments and colors, and they are swimming around at least part of the time. Their larvae actually are not as similar as you would think to other fish, but the adults are anyway very similar to what we think of as fish. There's no bones in these animals, but we're not actually sure if that's related to the fact that they've lost all their bones or if that's because bones never evolved in this group. We're not clear on that. Just because you have a primitive group today doesn't mean every characteristic it brings to the table is a primitive characteristic. Animals continue to evolve. They don't just get locked into time. They do have some of the characteristics that are ancient, but they also bring to the table new characteristics. So there are very few modern species of these, although there were probably relatively large numbers of species that look something like this in the past, right? Some worm-like animal. These animals start to get much larger. They vary in size from maybe 5 to 10 centimeters all the way up to a meter, right? So these are starting to get very, very large now. And they appear to have diverged from other fish groups about 500 million years ago. So these guys are relatively young, or relatively old groups, but they have a lot of stuff that looks like a fish. They really look like a fish. And I've already addressed this question: are these in fact a group? These are absolutely a group. There's almost it would be very hard to argue now that lampreys and hagfish are not very closely related to one another. Now let's talk a little bit about an enigma. So what I just mentioned to you before was the lampreys. They have some conditions which make them look like modern groups, but they don't have all of the conditions that make them look like modern groups. And one of the things that are not similar to modern groups is that those teeth were actually made out of the same material that your fingernail is. They're not really made out of bone, and that seems to be primitive for the group. It doesn't seem like that evolved and was lost. It really appears that they never evolved these very hard teeth that we have today. On the other hand, there are these animals called conodonts, and conodonts were really unusual for a long time. Conodont teeth are extremely common in the fossil record up to the end of the Triassic. Again, they actually go extinct at that point. But prior to that, you can actually date rocks because conodont teeth are so common. When you're in marine environments, you can just pick up different species of conodont teeth and know very closely to what age the rock comes from because they're everywhere. And conodont teeth are one of the sort of enigmas that challenged paleobiologists for a long time. What even were conodonts? Were they even animals? If they were animals, where did they belong? And if they weren't animals, were they plants? Or were in fact they not even plants? Were they just secretions of minerals within crevices of rocks or something that looks like a biological creation? That largely people disregarded that one, but it was very unclear where within the tree of life conodonts belong. It seems now that conodonts do belong within the maybe craniata. Maybe these might be more derived than lampreys. They're relatively small. The teeth are very small as well, but they do appear to have teeth. However, it looks like now the teeth that they evolved were evolved independently from vertebrate lineages that we think of today, and as a result, that would place conodonts further outside of the group of. They wouldn't be a fish anymore. They would move further out of that group and be, again, just right at that dividing point, right at the bottom. But what I want you to take away from conodonts, this is actually a very good reconstruction of what something like what a conodont would have looked like, is these are small animals. They're long and tubular. They're generally active in swimming in the water column. They may or may not have teeth, but they're probably primarily filter feeders. These animals may have also been able to take rips of flesh out of larger animals as well, and that's possible with the teeth that they had. So here's some pictures of conodont teeth on the left. Each conodont would have a group of different teeth, and each species would have an array of different teeth. Here's at the bottom is a picture of conodont teeth actually on top of a pinhead. So conodont teeth are relatively small and fragile, but you can digest them out of rocks with acids. We really are still unclear about where they fit within the larger tree of evolution. I've already mentioned that. They may be more derived, they may be more advanced than lampreys, but they may not be. It's not entirely clear. Teeth do appear to be built in a very similar way to our own teeth, but that seems like that was picked up separately. So the evolution of teeth arose, the way that they're built arose actually independently twice, which suggests the mechanism to build teeth is already present in primitive vertebrates, or primitive fishes, but they weren't using it to build teeth. They were using it for other things. Again, the real problem with stuff like this is when you have soft-bodied animals that suddenly appear in the fossil record at about 500 million years ago, how do they relate to one another? just looking at the teeth can be very difficult to necessarily understand how these groups are related because other groups don't have teeth to compare. So it can be very hard and challenging in that way to say, well, if this is a characteristic by which we identify these animals, and this is a characteristic by which we want to understand them, but other groups don't have them, how do they relate to one another? So more work is to be done on conodonts. I suspect that they will prove to be a very, very interesting group within the history of fish, but for the moment, it's not entirely clear to us where they sit within that tree. So if you read about them in a book, they'll often sort of hedge their bets and say, we don't really know, and they won't draw them on a tree because it's just too unclear at this point. I'm sorry. All of these animals that we've had have appeared basically at the very start of the Cambrian, in this very narrow bracket, the 520 to 540 million years ago in the Paleozoic. That's where we are looking at all these animals. You'll have noticed that these animals have some diversity, so they've split from one another. They've started to do different things clearly, but the body plan is very, very similar. They are, if you cut the heads off and just looked at the back of the body, they wouldn't look terribly different from one another. They're a little bit different, but they're not super different. Well, let's see what happens as we move into younger and younger rocks. These are still old rocks, but they're younger now. What begins to happen, what fish really start to do is they start to put on a lot of armor, right? So small, soft-bodied worms are very vulnerable to predation. One of the ways that you can prevent predation is you can start to armor up, and that's what a lot of primitive fish groups do. These are all jawless fish. PTERASPIDOMORPHI are all jawless fish, but they start to add layers and layers of armor, especially, and this makes sense, around the head, where the body is really vulnerable to damage, right? One hit on the area on your nerves, and that can be your death, even though the nerves make up a very small percentage of your overall body. Whereas somebody bites your tail, yeah, you can lose a lot of tail muscle and still survive. That can be regrown relatively easily. These look something like a fish, I guess, but they are really unusual and weird animals. I mean, look at this guy down here on the left. It's got eyes that are positioned at 90-degree angles to the front of the head, and what appears to be these nostrils. It's got this weird rounded body, and then it has this tail sticking out here that looks like a paddle. These guys are very heavy animals, very heavy, and that raises all sorts of questions about why would you evolve bones? Small amounts of bones do not provide protection, right? Having a very small, thin bone, which is how bone would have started to evolve, does not provide you with stability. It does not provide you with protection. And so bone probably doesn't evolve to protect the animals, but it rapidly gets co-opted into that mode of use. It's useful very, very quickly. Once you get some bone, it becomes very, very easy to get lots of bone, and lots of bone is really good at protecting you from predators. Predators at this time would have been giant sea scorpions and swimming arthropods, so animals that have big crushing claws and tearing mouth parts. And so if you can armor up, you can, even if they grab you and try to rip at you, they can't rip you apart. Or they can't rip you apart as quickly, and that gives you a chance to get away. What we think they did is they spent most of their time on the bottom now. They're still relatively active to some degree, but they're not up in the water column. They spend a lot of time on the bottom. They're probably inhaling a lot of bottom sediments, getting rid of the water, and then consuming the small amounts of organic matter that are in there, which may have been small worms, which may have been algae, which may have been just the organic matter, the sort of soup that is mud, basically. These guys are relatively small. You can see we're still down here in the centimeter range, so we're talking about animals that are less than 30 centimeters or a foot long, but they are acting more like a fish. They are starting to pick up things that are fish-like. Bones is very fish-like. Lots of armor is not on your outside of your body, but bones within your body that provide a lot of support and allow for much larger body plans are being picked up now. And it's not that these guys evolved bone and were like, oh, this will be great for getting larger. It's that they picked up bone, and that will be useful in the future for making very large bodies. This may be what we call the sister group or a closely related group to gnathostomes. There may have been a chordatespidomorph group that gave rise to the gnathostomes. The gnathostomes are jawed animals. The gnathostomes include all other vertebrates that we think about today, and they are the group that we are, right? We're a gnathostome. Not clear, though, and how you evolve jaws will be a whole other issue. How do you actually get to jaws? So here again is some of the diversity, some really cool stuff in here. There's an opening. Here's a relatively small opening, but lots of this thick armor. The cool things like long spears, that probably makes it very hard for an organism to handle it, so it's easy to drop or to release. These long projecting spines probably do the same thing, but they also probably provide some stability when you're swimming around. Some of these groups appear to have less armor than others, so they may have been more active in the water column. But none of these groups are probably particularly active. Even groups that are look like they swim around more often are probably too heavy to swim constantly. They probably have to come down to the sediment and pump water across their gills. Here's another group of jawless fish, and these appear to be a more active group. These are THELODONTI. These are jawless, but they do have scales. They've given up. They don't have that thick armor. Instead, what they've done is they've modified armor into a very small bone-like structures, into a very small form of armor, which is a scale. And that seems to protect the body while allowing them to be relatively mobile. When I say they're paraphyletic, that may mean that these are not an actual grouping. And that reflects our limited understanding of the fossil record of these guys. These may actually belong to many different groups of organisms that look something like each other, and so we dump them together. It's the same way as imagine if your uncle looked completely different from you, and I said, well, your uncle is not part of your family, and that's because he just looks different. Well, that's nonsensical. It's not good enough that he doesn't look like you, right? He is part of your family, very much so, and it doesn't make any sense to exclude him just because he doesn't look like you. That's just a coincidence in that way. It has nothing to do with your actual relatedness. Something else you're starting to see with fishes, though, within this group, as a good example, is they're starting to pick up lots of fins. Fins are starting to appear in these guys that are providing stability, so these guys are becoming active swimmers, right? They're really spending more and more time in the water. And you can see the time now. We've advanced in time almost 100 million years, right? That's 100 million years since something like a lamprey swam around, and this is what you get. It's different, but it's not stunningly different, I think. Evolution is a very slow process, and it doesn't necessarily work towards a goal. It doesn't set out to produce the best fish that ever existed. It has a group of organisms that are under selective pressures. Sometimes those organisms pick up traits that are more adaptive to their environment, and they succeed, and sometimes they don't, and they go extinct. These guys are all from shallow marine environments. And unfortunately, for right now, again, this is true of a lot of these primitive groups, we really don't know how these guys fit into the larger phylogeny or the history of fishes right now. So stay tuned to find out how we actually fit these guys in, and if in fact they are a real group, which they may not be. Now the Anapsida is another group of jawless fish. These guys are, again, extremely difficult to place within the history of fishes. They share a lot of characteristics with what we think are later fishes. So we want to place them very closely to the branch between jawless and jawed fishes. But they have other characteristics which are very, very dissimilar from what we think of as modern fishes. What you'll see here again is this is probably convergent as well. We have scales and armor being reduced to allow the fish to be mobile. So fishes have two options basically. You can either armor up so that when animals grab you, they can't eat you, or you can armor down and try to be very lightweight and get away. Use that muscular body to get you away from that predator animal. And you can't do both well. You can't be heavily armored and be fast. You can be heavily armored, but you can't be fast. You can be fast, but you can't be heavily armored. And that's something that fishes have been dealing with now since they have evolved. Or since bone evolved, I guess, is really where they've had to split the option to do either one or the other. And that is led to this pickup or the loss of bone in different groups. And we'll see that in the future. They do have a single nasal opening, which makes them appear to fall very close to lampreys, which also only have a single nasal opening. It turns out that two nasal openings, like you have, where you breathe in through two nostrils, is actually a derived condition. All fishes start out with a single nasal opening that splits into two nasal openings, and that's what we have today within our own nasal opening. But lampreys have the primitive condition, which is a single nasal opening. These guys, again, they are really starting to get fins here, so they can be up in the water column and control what direction they're swimming and how they're actually swimming. There are folds on the underside of the body now that provide stability, so the animal's not constantly rolling around within the water column. It can hold itself up. And again, the timing here is about 430, maybe to 410 million years ago. We don't know. We're hoping to place these guys in the tree of life in the next 10 to 20 years, but it's hard to guess right now. We need more fossils. We need more scientists looking at these groups and really starting to peel apart what makes this group special, what conditions does it share with other groups. And that will require new techniques, but will also require the modern techniques, like things like CAT scans, get well applied to a lot of these different fossils, so we can get a really good understanding of how these groups relate to one another. Here's yet another group, the Galeaspida. These also lived from about 430 to 370 million years ago. Very weird groups, right? Look at this mouth. What a weird mouth that is. It's not a jawed mouth, it's just an opening. This group appears to have no paired fins, so we would say, it doesn't have the paired fins that we see in later fish groups, and it doesn't really seem to have the diversity of fins that we've seen in other groups, so it's probably more primitive. But there is this very interesting feature. The first gill arch, which is the gill arch that supports the first gill, obviously, has a facial nerve that reaches it and it innervates it. And that may not seem like much. That may seem like sort of a trivial detail. But that's actually really important because facial nerves innervate very specific gill arches, and they do it so specifically that if you pick up any vertebrate animal, you can number the same facial nerve in every single animal because they go to the same places and they start in the same location in the brain. So if this animal has that nerve, that suggests very strongly that it's actually closely related to our own group. But it doesn't necessarily mean that it has to be. But again, we really don't know. These guys all appear to be relatively sedentary bottom dwellers, so these guys are into the armor thing, right? So you can see a lot of armor around the head, and then a relatively mobile tail that allows them to move around. But the head is heavily armored, so these are relatively sedentary animals. This animal is very exciting for a number of reasons, but one of the reasons is it really doesn't look like it. Really doesn't look like that thing has any characteristics that look like a jaw. But actually, when you go in and look at the fossil, it's actually got some of the characteristics that make true jaws. It's the evolution of jaws happens very slowly, but what you can see is that the arches start to form, and they will soften in the middle and allow the mouth to be closed and opened a bit. And at some point, that becomes a really active opening and closing of the mouth. And once you have an active opening and closing of the mouth, you can layer in teeth, and now you have a phenomenal biting apparatus. But animals like this actually give us real insight into how jaws evolve, even though weird moon-eyed fish with giant hole in front of head for mouth doesn't seem like it would provide any information about that. This is a very famous group of fish. This is the Ostracii. The Ostracii are actually relatively well studied, at least this species down here. There was an individual in Germany who studied this fish in the late 1800s, I believe. And what he did was he had specimens, and he would sandpaper the specimen down, and he would sandpaper down by millimeters and then draw exactly what he saw. And what he ended up doing by the time he sandpapered the entire specimen away was he had a 3D reconstruction of the entire fish from the inside and from the outside, right? He actually could see things like the fossil brain preserved and draw the nerves on it, right? And what's very interesting about the Ostracii is when he did that, the brain was set up in the very same way we see future vertebrate brains set up. So that's already locked in. And so this is a very, 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 very cool thing. Today, we wouldn't need to destroy specimens to do that, although at the time you would have had to. We can do things like CAT scans and basically do the same thing. But that kind of work, which took literal years to sandpaper down a single specimen, is very, very important, and it provides really important information. This is a really interesting group, right? I think people would have no problem calling these fish. They've got clearly paired fins, right? There's fins out to the side that allow the animal to really tightly control movement now. It appears very, very fish-like. There's a very clear caudal fin, right? A tail that's actually pushing you through the water column. They've got a clearly defined dorsal surface with a dorsal fin that's helping the fish prevent itself from rolling. They almost certainly have a very strong electrical organ around the head. That's what this pad is. It's extremely well innervated within the fossils. That's probably for detecting small things like worms or prey items within the mud that it's swimming around in. And these are becoming very large now, right? I said before that lampreys were up to 1 meter. Well, these guys are also up to 1 meter. We're starting to pick up the size increases in fish. Now that things like bones have evolved, you're able to provide very strong support. Fishes are getting larger and larger. One of the things about Ostracii is they're actually pretty rare, and they stay rare. They evolve. They're rare in the fossil record. They stay rare, and then they go extinct relatively shortly after they evolve. So they're about 60 million years. This is a group that in that sense, I guess, failed. But if the characteristics that made them successful were passed down in other lineages, then that may not have that actually may not be true. Their lineage may have survived in some sense, but the group itself is now extinct. And so that's going to end the jawless fish, the anapsids as a whole. Anapsids, again, not really a grouping, so I'm not going to use that word very much. But I will say that we can say jawless fish, all the fish that don't have jaws. And what you're going to see now is we're going to really start to talk about jawed fish. And jaws are huge. Jaws, although bone is important to getting large, jaws are really important to getting large too, because to feed a large animal, you need to take in lots of high quality food. And to take in lots of high quality food, you need to be eating animals, big animals, and lots of animals. And you're not going to get away with eating lots of little things unless you do a huge amounts of filter feeding. Huge amounts of filter feeding. And the diversity of large filter feeders available to you is relatively low, right? But jaws really, really, really allow you to do that. And even today, the largest filter feeders are not jawless fish. The largest filter feeders are jawed animals. There's a real advantage to being able to close your mouth when you're swimming to prevent yourself from having to push a lot of water through your mouth when you don't need to. So this is a primitive, relatively primitive jawed fish. We're going to talk about him in the future. It belongs to the group called the placoderms. You can see a ton of armor, and this kind of jaw is amazingly powerful. And then this, of course, would be your characteristic, I think, jawed fish today, right? Something like a shark. And, of course, we'll talk about sharks as well, because these are also another group of jawed animals that are very important. So with that, I'm going to end it. And if you have any questions, leave them in the comments.